sterile medium. Taking bacteria from a culture, transferring to a sterile medium. That's inoculation or subculturing. And that's done aseptically. No proper labeling techniques, so you need to know the 5 different parts of the labeling. Been over that. And Petri plate placement for incubation. Also, as far as labeling and the Petri plate, you should know that the labeling is done on the bottom, right? Not on the lid, on the bottom, around the edge. And then the Petri plate's put in for incubation, inverted, right? Upside down. So the lid should be always be on the bottom. Know what the typical incubation temperatures we use. 25 degrees Celsius, which is room temperature. 25 is room temperature. 37 is body temperature. You should know those as each, as body temperature and room temperature. 37 body, 25 room temperature. Be able to identify and name the cocci and bacilli bacterial shapes and cellular arrangements. So just know, if I have I think I mentioned, you're not going to need to look at any slides under a microscope. I'm going to have photos. So if I have a picture of round cells, those are cocci. If they're rod-shaped, those are bacilli. If they're in clusters, it's staph. If it's a chain, it's strep. And then there's spiral, but I won't show you that one. Know the gram staining steps. So you need to know crystal violet as the primary stain is the first step. Quiz material again. Crystal violet primary stains first, then iodine is the mordant second, alcohol, decolorizing third, safranin, counterstain fourth. Those are the steps, and the color of gram positive and gram negative. Gram positive is purple, gram negative is pink. Know the names of all stains that we used and which staining procedure they were used for and why. So the gram stain, we just went over that, and you need to know which one we used for that. And then we just used carbol fuchsin and methylene blue. There's tons of different stains. We used carbol fuchsin and methylene blue for simple staining. That's the staining procedure that I'm referring to, simple staining.
Do you know the colors?
Yes.
Oh.
You should be able to spell these.
Is the test going to be multiple choice or?
No, it's a recall. It's the vast majority of questions are recall. There might be 3 or 4 tops, if that, multiple choice. So most of them are going to be like one-word answers. There's no word bank or anything. Recall, and maybe you very, very, very short answer, like 3 or 4 word short answers. Nigerson, we did not actually use it, but you should know that it's used for negative staining, which I'll mention in a second. So that's for negative staining. Nigerson.
And gram staining is simple staining.
Gram staining is a differential staining, which I have not mentioned that term, although you've had it.
Yeah.
That's differential staining. Know that electrically positive stains are basic and electrically negative stains are acidic, and the basis for how cellular staining works. Talked about this already. The basis for how staining works is cells are typically negatively charged, and then if you have a positively charged stain, like carbol fuchsin methylene blue, they are attracted to the negative cells. So opposite charges attract is the basis for that. Positive stain, negative cell. Okay?
So basically it's like, can you repeat that? Sorry.
Negative or positively charged stain binds to negatively charged cell. Let me jump down to negative staining right here. Unless it's negative staining, in that case, nigrosin, which is negatively charged, stains the background. We'll talk about this in lecture. So negative staining is the background is stained, because you're using a negatively charged stain. Like charges repel. So the cell itself is not stained. Know the difference between simple and differential staining. So we've been over this. Simple staining is one stain to see shape and arrangement. One stain to see shape and arrangement. Simple staining. Differential staining, two or more stains or dyes. Two or more dyes to differentiate cell types. For example, gram staining. That's a differential staining technique.
One more thing to differentiate.
Cell types, such as gram staining. And then the negative staining I went over. Acid-fast and spore staining, we did not do it. So you don't need to know the details, just know that acid-fast is for mycobacteria. You know that already. And you should know a couple of examples of them from quiz material again. Mycobacterium tuberculosis causes tuberculosis. You take a sputum sample, for example, and do a slide of it. You can see it. Or Mycobacterium leprosy. And then the spore staining for bacillus or clostridium. That was quiz material also. Bacillus or clostridium genera form spores. Quiz material. So again, we didn't actually do that, but we just looked at the pictures in the book. Know what selective media, differential media, and enriched media are. What does fastidious mean? Selective media inhibits certain microbes, allows the growth of others. Selects for certain microbes, inhibits the growth of others. You can put it different ways. Differential differentiates between different microbes. I'm going to define that right now. Enriched media can be used for lab. Let's just think of it as enriched media is used to grow fastidious microorganisms, which means they're picky eaters. They need special nutrients. So enriched media can be used to grow fastidious microbes, the picky eaters. Know each media type. Here's a lot of information within this about this stuff. Know each media type, MSA, and so on. So now, for the quiz, I think I let you go whether you gave me the abbreviation or the full name, but for the lab, make sure you know the abbreviation and the full name of the media. So MSA, for the lab exam. So MSA, you know, is mannitol salt agar, PEA is phenylethyl alcohol agar. The Columbia CNA, you just need to know it as that. There's no abbreviation. CNA happens to be an abbreviation for something. You don't need to know the full name of that. It's colistin, malolactic acid.
What did you say about MSA and PEA? You want us to know those, right?
You need to know the full names of all of these acronyms,
Okay,
except for CNA,
okay.
so you just go back in the book if you forgot, back in the lab book. And then see table below, which I'll get to in a second.
So basically just know everything you have enough of those for those?
Yes.
What they inhibit?
Yes, and a little more, which I'm going to talk about. It's in this table that I'm referring to here. So I'm gonna spend a bit, quite a bit of time on this. I'm not leaving it yet. Let me read this through, and then I'm gonna go to the table. So know each media type. In other words, what they're used for, and what organisms may be inhibited by and selected for. Be able to identify certain bacteria growing on a selective or differential media plate. For example, E. coli on EMB, S. aureus on MSA. So those are the two I told you in lab, remember? Those are the only two species names specifically that you need to know what they're doing and on what plate, what media. So when I say be able to identify certain bacteria, these are the ones right there. So start memorizing all the other results if you've written it down. Let me continue reading this here. Related to the above, know the selective or differential agent, chemical, basically, in the different media types which allow for the growth and identification of the different bacteria. So all that information, all these things that I want you to know, you're going to get all of that from this table, which is the second page, this table, and this table, which is largely redundant, the two tables, but there's some slight differences. So let me go back. So you just basically have to memorize it. You just got to memorize it. And I'll just go through a little bit of it. So MSA is mannitol salt agar, whereas on the other table I actually spell them out. Mannitol salt agar, the pH indicator is phenol red. We've been over this. The selective agent, high salt concentration. Remember brackets means concentration. So what is it that inhibits? I'm saying, what is the agent that inhibits? Salt. What is the agent that inhibits in PEA? Phenylethyl alcohol. What's the agent that inhibits in Columbia CNA? Colistin and malodixic acid. I just mentioned it a little earlier today. I never mentioned the actual
So that means that
agent
it
lab.
won't grow there?
I'm sorry, say again.
That means it won't grow there. If you're saying
Right,
it
this
inhibits-
is, right, so we're talking about selective media, which inhibits certain microbes and allows the growth of others. So what is it in the media that is inhibiting
The growth of that word.
the growth, of that particular organism? What's inhibited in MSA? What species is it, what microbe is inhibited in MSA? Non-halophiles. What's the agent that inhibits? Salt, high salt. What's inhibited in EMB? What microbe? Eosinmethylene blue.
E. coli.
Gram positive. Right, gram positive. Gram positive. E. coli is differentiated. Remember with the metallic gene. So what is the agent that inhibits the gram positives? Eosin and methylene blue. Calling it the selective agent. It's involved in selecting against certain
So-
microbes.
Columbia would inhibit gram negative, that would be the.
Right. As you remember from the quiz, PEA and Columbia CNA are the only two that inhibit gram negative. And you need to know that stuff, which is over in this column. Before I get to that. The differential agent and property. We talked about this also. MSA, mannitol, is what's utilized to be able to differentiate. That's the actual agent, the chemical, and the property is fermentation. So it's just memorizing the stuff. Okay?
So, okay, so in MSA, so the start area, it fermentates and turns yellow, the lactose fermentates and makes it yellow.
Phenol red,
Phenol.
mannitol is fermented and the acid is produced and phenol red turns yellow for that particular one. Right, the pH indicator is listed right there. And so then what do you get? What are the results? As you're talking, Staph aureus yellow, Staph epidermidis is red. So they grow, but one's yellow, one's red. What's growing on PEA? Gram-positive. What's growing on Columbia CNA? Gram-positive. Height slash, what is it differentiating? What's the property hemolysis, gamma, alpha, and beta? Differentiating those. EMB, what is growing? Gram-negative enterics or coliforms we can think of. Remember I talked about that, enterics and coliforms? Remember, you know those terms. Make sure you know those terms. Enteric, gram-negative intestinal, and the coliforms. There was a list of characteristics that I gave you in lab that you wrote down. And then what does it look like? Pink, pinkish color, if you recall. Anything that was pink, we would consider lactose fermenter, because that's the differential agent, lactose and fermentation property. So you have to think about this as you're studying through it. These are basically what the result tells you. E. coli is a metallic machine. A non-coliform, there would be no color. If it's growing and it doesn't ferment lactose, there's no color. Right, you have to go back. I'm not going to go into the nitty-gritty details again. We went over all this. You got to go back into. If you're not remembering this, you got to go back into your notes and look and clarify it. It's still not clear that you can ask me. Okay, so it's basically memorizing this. I've been over all this. Get over these plates. Now let me go back to this other table and read through this. So I told you this is mostly somewhat redundant. So it gives all the media, plate media test. Now here I'm saying what is actually inhibited, specifically, so you know. So it's really easy to remember what's inhibited. You got the one outlier, if you will, the MSAs, the non-halophiles, and then PEA, Columbia CNA are the only ones that inhibit gram-negative. The other three inhibit gram-positive. That should be easy to remember. Microbes isolated, Staph aureus, Staph epidermidis. Gram-positives isolated. Now cross off here. I won't specifically refer to this. Cross off staphylococcus, streptococci, and enterococci. Just want you to know that it's what it inhibits and what it selects for, and then what it differentiates. Here I wrote distinguishes, differentiates, same thing. It's differentiating hemolytic species, alpha, beta, gamma. So that's your alpha, beta, gamma.
So when you put microbes isolated, you're saying what's growing?
Yes, yeah, exactly. What's growing on the plate. Cross off staphylococcus, streptococci, and enterococci. Eosinophilin blue, microbes isolated or that grow on the plate. Coliforms, non-coliform enteric. So coliform and non-coliform enterics. I should have put S there, enterics. So because coliform, you can have enterics can be coliform or non-coliform. And specifically E. coli.
So what did it say that one more time?
I was just saying, I put here, if you look at the board, I have here enteric singular. I should have put it as plural, because they can coliform and non-coliforms are both enterics. And remember, and you need to remember to tie things together, so, what else do you need to know about this? And this is what I think I was talking about, active studying. They're talking about that with you, as opposed to passive studying. No? I always talk about this with my biology 1 class, because usually students in that class are a little bit earlier in their studies. You guys have had a few more classes, but still, it's good to get tips on studying techniques. Active studying, as opposed to passively studying is reading through that and just sort of trying to memorize it. Active studying would be, maybe writing it down. That would be, that would be really great to be able to, you know, I personally, to be honest, maybe I did that a few times in college, just 'cause, you know, got lots of stuff to do, but some people do it regularly. It really helps. Also, active studying is asking yourself, what else do I need to know about this, right? So that's what I was just referring to. Ethanol, methylene blue, I'm giving you some information about that, but there's other things you need to know about this, right? So don't just go back to the other page and read it. Think about, okay, what else do I need to know about that? And hopefully you'll think of lactose fermentation. You know, we're differentiating lactose fermenters from non-fermenters, which is what sort of ties into the coliform and the non-coliforms. Coliforms ferment lactose. That's active study, a type of active study. You're forcing yourself to try to think about it, and that burns the memory into your brain instead of just reading through it. Trust me, you'll get results on the test if you do that. Let me continue on. Any questions? HEA, hectoenteric agar, differentiates Salmonella and Shigella, we know that from quiz, it's straightforward, species, from enteric lactose fermenters. Now here, we need to change lactose fermenters to sugar fermenters. That's how I refer to it. Lactose is one sugar out of 3 in HEA. So, and I didn't phrase it in terms of enteric sugar fermenters, but that's how we can think of it, because we're just growing gram negatives, right? The sugar fermenters. And then McConkey, coliform, and non-coliform enterics. And again, lactose fermenters, non-fermenters, remember that from the quiz. So, gotta memorize this stuff, it's the only way around it. Know how they relate to each other. Any questions about these media? Any questions?
So in the stations, you might have a plate, basically.
I won't have a plate, but I'd have a picture.
That
And I'll show you from the PowerPoint.
Okay.
So I'll be referring to the stuff again with the PowerPoint.
Okay.
Yeah, I decided not to have living microbes on the test. Just, accidentally, if somebody accidentally drop it or something, spill it. Okay, so that's relating to this stuff, the table, and again, I'll go on to the PowerPoint and talk about this some more. Understand the terms, so just know the terms. Halophile, cyclophile, mesophile, thermophile. So cold-loving, moderate temperature-loving, hot temperature-loving microbes. Acidophile, they grow in low pH, neutrophile, around neutral pH, alkalophiles, high pH or basic pH. What is pH? Hydrogen ion concentration. That's what pH is. Hydrogen ion concentration. Reach the H plus concentration. H plus concentration. And the different terms describing oxygen growth requirements. We've been over that a couple times, so I'm not going to go over it again unless you specifically want me to go over something, but that's the facultative anaerobe, obligate, aerobe, all that stuff.
Where would we find that is that in our lab book?
It's in your lab book, it's also in your lecture notes. Remember the picture of the test tubes? I don't know if you were here, the picture of test tubes. Just went over it recently, chapter 6? Yeah, chapter 6. It's in there. And be able to identify a culture based on its oxygen requirements. So here, I'm not going to have an actual culture sitting there, but I might have a picture of something with, let's say I had a picture of a test tube with obvious growth only at the top and not at the bottom. It would be an obligate aerobe. If we're talking about a fluid thioglycolate medium. Remember the FTM, the fluid thioglycolate? That's where we grow that kind of stuff. Or I can describe it just in text I could say, you observe a test tube and there's growth throughout the test tube. Facultative, anaerobe, or aerotolerant. It could be growing throughout. Okay. Understand the basis for and what a positive result looks like for the milk, tributylin, and starch agar plates. The starch agar, remember we added iodine, and the iodine binds with starch. So if there's, and turns almost a black color. So if there's no black, if it's clear around the growth, that's positive. That means that there's no starch there to react, which means it was broken down, which would be a positive result.
Say that one more time.
For the starch, there'd be no color around the growth after you add the iodine, which means starch is broken down.
So if there was starch and the positive result means that there's no color.
Right, around the growth. And there's a picture in the book. Right. Tributylin in milk, there's just a clearing around the growth. It's positive. A clearing around the growth. It's positive. Also study all material from the PowerPoint lab midterm review slide, which I'm going to go to right now. That's for that. Any questions on what I've covered so far? Okay, so let's go to the PowerPoint. So I will definitely have pictures directly from the PowerPoint on the test. So you need to go through all of these and memorize basically the photos and all the text that goes with it. So for this one, and some might have questions on it without answers, just to get you to actively study, basically. So gram-positive staphylococcus notes. Note purple, spherically shaped clustered cells. So these are gram-positive because they're purple, they're cocci, and they're staph because they're clusters. Gram-negative bacilli, you can see they're rod-shaped, they're pink. They're not in clusters or chains. Acid-fast staining. So from know all the text, mycobacteria are identified. And typically, and I went over this also in the lab, but typically we use a pink color for the primary stain to show the mycobacteria. So in this case, the pink cells would be the mycobacteria, and the non-mycobacteria, or non-acid fast, would be the blue. So you should know that. I can put that picture up and ask you which ones are the acid fast positive, or the mycobacteria. I could put this up and say which letter are the spores or endospores. That's pretty easy because they're going to be smaller than the vegetative cells. What you're looking at here are vegetative cells, all the pinkish, reddish ones, and all the turquoise are spores or endospores, produced by either bacilli or Clostridia, bacillus or Clostridia. Here's your milk agar plate. Just talk about this. So clearing around the growth is positive. Nothing much you need to know about this. Casein is the milk protein. So casein is broken down. You get this clearing around the growth. These are negative. These are positive. Don't memorize the species. Starch, as I just talked about, no color around the growth is positive. Because all this dark purple-black is where the iodine reacted with the starch. So here, obviously, the starch is broken down because there's no color there. Positive for the enzyme to break down starch.
So on the left, there's no color.
Yeah, this would be negative because starch They're not broken down. PEA inhibits what? Gram-negative. Inhibits gram-negative. B, there's no growth. You have E. coli there. That's gram-negative. Staph aureus is gram-positive. You see growth. Why? Is this media selective or differential? It's selective. Why? Because it's inhibiting the gram-negative E. coli. Your active studying, here's another good example of active studying. What inhibits gram-negatives on PDA? Don't go to the table. Phenylethyl alcohol. Can't remember it, then you go back to the table. So you try to recall it. That helps form the memory, the learning. Learning is largely just memories. And so on. Gelatin hydrolysis. We didn't actually inoculate this. We will in the second half of lab. But I just wanted to make a mention again of why we use agar instead of other things. Gelatin can be used as a solidifying agent, but it's used for testing purposes. So just again, as I talked about in lecture and in lab probably, why do we use agar? Why is it superior as a solidifying agent? Number one, most bacteria are unable to digest agar. That's one reason why we use it as a solidifying agent. The other reason, number two, it remains solid well above room temperature. Those two things. You don't need to memorize this picture because this is gelatin. I just want you to know about the agar. I'll show you this again for the final lab exam when we actually use gelatin. McConkey agar. So, I'm not going to read through all of this, but this is information you're going to need to know. Try to answer the questions. You should be able to answer them. Look it up. If you can't find it, if you still can't answer these questions from these slides, then you can contact me. So looking at the pictures, this compared to this one here, it's pinkish color. So what does that mean if you see a pinkish color? It means it ferments lactose. We talked about this in lab when I was going over this. I know that's a lot of material to remember all of it. If it doesn't turn a color, then it's not fermented lactose. And so on. What is the pH indicator in this media? Try to remember it. If you don't remember, after thinking about it for a while, go to the table. I have it listed on the table.
It doesn't turn color, it's not fermented.
Correct. Okay, so you read through this. Eosin methylene blue. Again, it's lactose fermentation. The metallic green sheen on the plate characteristic of E. coli, which we talked about, and I think you saw that somewhat. Is EMB a selective or differential media? Remember, it's both. Quiz material. What are the dyes in EMB? The dyes are eosin and methylene blue. Those are actual dyes. Eosin is a dye, methylene blue is a dye. They happen to be the selective agent as well. If you go back to the table, you'll see that. That's what inhibits gram what? Gram positive. Yeah, so there's a lot of ways of doing active studying with this stuff. There's a lot of material, but it's all there. It's just a matter of getting down into it, finding the time to study. Yes?
All the media plates are both selective and different besides
Well, let me just, so I don't make a mistake, you actually have to go back. Let's see if we get the other one. And then, so you asked if they were all selective and differential except for PEA. MSA is selective and differential. This one is just selective. This one is selective and differential. Selective and differential, so correct. And that's a good way to memorize things. Yes, you can group things like that to memorize them. Okay? Okay, so I'm not going into, I'm not reading through every one of these slides because we've been over it. MSA, now this is a pretty clear cut one. We always have beautiful results in lab. You probably remember this just by looking at it. I've also talked about it in lecture. Why does SRAs produce a yellow color?
Fermented.
Meant what?
Vanilla red. Because it's a hollow bar.
Hint.
Mannitol.
Mannitol. Mannitol is the sugar, not lactose. That's another one. So it ferments the mannitol, produces the acid, changes pH, phenol red turns yellow.
So Greg, you asked that question, did you say fermented mannitol?
It depends on how I ask you. If I said, what is the sugar that's fermented? Mannitol. I could ask you different things about it.
So if you say why does this always produce a yellow color because it's mannitol? Or you have to say
yes,
something
that
about
would
the
be.
acid as well?
Well, that would work. And for, I'm glad you asked the question. You're not going to have a lot of space to write, so I don't write the questions in a way that requires a long answer. So to be a full, accurate answer, if I ask why is it turned yellow, that's more than would fit in the space that I need. So if you just said it ferments mannitol, that would be acceptable. If you said it changes the pH, that's acceptable too. There might be more than one acceptable answer. Blood agar, so just a different hemolysis, gamma, no blood cell destruction. Is blood agar selective, differential, or enriched? It can be all of the above, but for our purposes, for Columbia CNA, it's all of them. But I don't want you to think of that of the Columbia CNA, because that's a special one. Blood agar in general is differential and enriched. Okay, just regular blood agar is differential and enriched.
It's not selective.
Correct. It has to only selected if special ingredients are put into it, like CNA, which is colistin and malic acid. Did we do the throat swab yet? Okay, so that was just regular blood agar, and we also put the antibiotic on. That was regular blood agar, selected and enriched. It wasn't differential and enriched, not selected. So gamma,
It's enriched because of the blood.
it's enriched because there's partly because of that, and just there's lots of it's just chock full of nutrients. So pretty much anything can grow on that, or lots of things can grow on it. The alpha hemolysis, remember, is partial red blood cell destruction, and typically gives this greenish color. As you can see, obviously gamma, there's really no discoloration to speak of. It's the color of the growth. And then beta, complete red blood cell destruction, and then you have a clearing around the growth, obviously different than alpha. The end. So you got three Word documents, two separate documents, one with 2 pages, one with 1 page, 2 tables, and then the list, and then you have the PowerPoint, all that to study.
Gamma is no change, right?
Right, gamma is no hemolysis. Any questions about the lab exam?
But the answer didn't get qualified back to you. What is
Did you write it down from lab?
I'm looking for
It's their lactose fermenters, their gram-negative rods, they're facultatively anaerobic, and they produce acid and gas. All those things that you probably wrote down in the lab. I wrote it on the board. I actually wrote the whole list on the board. Facultatively anaerobic, gram-negative rods, lactose fermenters, produces acid and gas. Those are columns. Any other questions about the lab exam? Okay. Remember, you can always come see me during my office hours. If you email me, please don't email me a whole list of questions, because I won't answer all of them. I get lots of emails. So if you have lots of questions, you need to either come see me during my office hours, or you can call me during my office hours. It's hard for you to get here personally, and I always pick up my phone unless I have a student with me or if I'm not in the office. If you leave a message, I'll call you back as long as I'm able to with the Valencia connection. Occasionally, for some reason, I can't, depending on the person's phone number, I can't get back to them. Okay. So that's going to be Monday. Let's just say tentatively now it'll be at 10 o'clock. But I'll confirm that in lecture on Monday when we actually will have the test. And you don't need anything except a pen or pencil. I'm going to supply an answer sheet for you. No scans on.
So we have a midterm lab exam and then we have a final lab exam?
Well, I think they call them that, but it's actually just two separate exams. They're not two.
They're not comprehensive, yeah, they're not.
Yeah, the last one is not comprehensive.
Okay,
That's just gonna cover the second.
okay.
Okay, is everybody in here? Did anybody leave? We're all in here, because we're gonna have a presentation, I don't want disruption. Okay. Carolyn? Oh, hold on.